what do I mean for functional data. Okay. So when we do statistical analysis, we can have univariate variables. So in this case, basically for each subject, we measure something, so some measurement of interest, and we can do a lot of statistics on that. We can have a bit more complex version that is multivariate statistics, in which for each subject, we actually have a bunch of variables that we measure. So the case of functional data is the infinite version of it. So basically, for each subject, we measure something in a continuous time. You can think about this curve. For example, this can be some continuous glucose monitoring curve that can be a functional data. So by functional data, I mean any type of data that is measured over a continuum, and in general, it is smooth. So it has some smoothness on this curve. Functional data analysis is the statistical analysis of functional data. So the smoothness is an important characteristic of these curves. By the way, it's called functional because curves are mathematical functions. Okay, so it doesn't mean functional in any other meaning, just because of that. So here we see two examples of two sets of curves, and you can see that one characteristic is that, at least in this example, they are pretty smooth. Another characteristic is that usually functional data are pretty complex, so we cannot really describe the pattern that we see with an easy formula. So the idea of functional data analysis is to try to exploit the shape that we have in this data to gain insight on this data and to really analyze this data. So in practice, we have a lot of problems with this data. First problem, well, we usually observe this curve with a lot of noise. So here there is an example of Vancouver precipitation data. So on the x-axis, we have the day of the year, and we have the precipitation on the y-axis. And you can see that there is some trend, but there is also a lot of noise. So one of the issues we deal with is how to deal with this noise and how to filter out some of this noise. Another problem that we can have is that the data and the curves can be measured at non-equally spaced points. So for example, here we have growth curves for children. So we have age on the x-axis and the height of these children on the y-axis. And we can see that we have a lot of measurements at the beginning for babies, basically. And then we have measurements only once every year. And then at a certain point, we start having measurement every six months. We can have also way more complex functional data. For example, we can have that these time points in which we have the measurements actually different for each subject. So each subject has his own observations on his own time. And also we can have cases in which well, we actually have only a few points for each subject. So we need somehow to recover the functional data starting from that. In the epidemiological and longitudinal studies, well, we have a lot of examples of functional data, even if usually they are not treated as functional data. In this figure, you can see an example of disease progression over time. So these are data about Parkinson's disease. And basically on the y-axis, we have data for a lot of subjects. So all these gray, let's call them curves. They are pretty rough and pretty noisy. But those are functional data, okay, with all the problems that I was telling you. We can have also continuous recorded data. These are perfect examples of functional data. So here we see activity counts done by an accelerometer. So we have the numbers of steps for different time of the day for two groups of children. So here we have, we have the difference between warm and cold days. And here we have the differences between children with asthma and children without asthma. And one last example, we have again a curve in time. And the measurements in this case are the glucose level measured in continuous time. Okay, so I told you that the first problem is that we have a lot of noise and we need to somehow filter it out. We also have another problem that is we usually do not really observe the functional datum. We do not really observe the curve. What we have are just some points. So the first step is always to use smoothing to remove some of the noise and recover our functional data. So this is like the first step before we can do any type of analysis. Another problem that we have with functional data is that sometimes the time is not aligned between subjects. So one thing that we often do is registration or alignment of functional data. So here you can see two examples in which we have these peaks. These are ChIP-seq peaks, but doesn't really matter. And we do an alignment using shift or using shift, but also allowing for scaling, so stretching on the x-axis. Okay, so once we have our functional data, what can we do? Well, basically all the methodology, all the statistical methodology you can think about for multivariate data, you can find them also for functional data. So there is almost always a version for functional data. So for example, we can have a functional clustering to find groups of curves based on their shape. We can also do hypothesis test. So for example, if we have two groups, we can test for differences between the two groups, looking at the shape of the curves. Or we have regression model to try to understand the correlation between different set of curves or set of curves versus some scalar measurement. Okay, so in this talk, I actually want to show you a bunch of applications. One big application to biomedical sciences and some application to omics sciences. In the case of omics sciences, it is not obvious what I mean by functional data because here we don't really have time. So we don't have longitudinal data in this case. But in this case, the curves are curved over the genome. So the x-axis are the nucleotides or windows that are one after the other along the genome. And we can have different type of measurements. Like in this example, we have substitution rates, so mutation rates. But so let me start with the biomedical science example, because I think this is like probably closer to the research of the audience today. So I'm going to talk about an analysis of COVID-19 data in Italy. So in this case, the curves we are interested in are mortality curves in different regions of Italy. So this part of the talk is based on a work with a team of Italian researchers. So Tobia Borchi was a PhD student in statistics at Penn State University. Jacopo Di Iorio is currently a postdoc at Penn State. And then we have Francesca Chiaromonte, who is a professor at Sant'Anna School in Italy and at Penn State University. And Lorenzo Testa was at that time a master's student at Sant'Anna School. So we're all Italian, so we decided to study what was happening in Italy in 2020. And actually, even if almost all these researchers are based at Penn State, so in the United States, they were stuck in Italy So for different reasons, they were in Italy when everything began in February 2020. So they were stuck there. They couldn't go out. So basically, this project was like going to therapy a little bit. So having, meeting every week and try to see if we could, do some science out of this very bad disease. I was the only one outside Italy, so I was here in Canada. So I actually didn't live the very bad months in Italy. So here it was much better, actually. So let me start with some timeline. So in Italy was basically the first country outside Asia to be hardly hit by COVID. So the first cases, we had the first cases at the end of February 2020, first non-travel related cases here in the north of Italy. So in the Lombardia and Veneto, so those are the two regions where actually everything started. And the situation became bad pretty quickly. So at the beginning of March, there was a nationwide lockdown, and the 23rd of March, all activities were suspended, and basically people could not go out of their house. Well, COVID-19 in Italy hit very, very hard. So only during the first two waves, that basically ended here in the summer 2021, we had 126,000 deaths due to COVID. These are at least the official data. Then in reality, there are way more than these. One striking aspect about this COVID-19 in Italy was that in different regions of Italy, actually the epidemics was completely different. So the result of the epidemics was very, very different in different regions of Italy. So in this figure, you see the official mortality curves. So it's basically mortality per 100,000. 10,000. Well, yeah, 100,000 people. And the different colors are the curves for the 20 different regions in Italy. So, well, we can see that there is a huge difference. If we look at the first wave, there were some regions, especially in the south of Italy, that basically didn't have COVID. So they were in a lockdown, but they had basically nothing. When we had some regions like Lombardia and Valle d'Aosta in the north that were hit very, very hard. So the goal of this analysis was to try to characterize the patterns of COVID-19 death across the Italian regions and try to see if there was any explanation for this heterogeneity among the regions. So the first problem we encountered was about the data. So we had the two sources of data. One was the official count of COVID-19 death from the Civil Protection Agency of Italy. The other one, we computed the differential mortality between 2020 and the years before. So in this case, we're considering the entire death. So we're really looking only at the differential mortality, so the excess mortality that we have due to COVID. In these two figures, you see two examples of the two regions where actually everything started. So the dashed line are the official data, and the solid line are the differential mortality data. So, I mean, we can see that at least here in Lombardia, well, the difference is huge. So there was a dramatic undercounting of deaths due to COVID. This is true at least in the first wave. The problem was that many people were just dying at home without a test. So they were not counted as COVID death, but I mean, they are. it's pretty obvious that they are. But the problem is that we have a problem also using these differential mortality data. And the problem is that these data can fluctuate also to negative values. So we can see, for example, here at the beginning of February that the differential mortality was actually negative, and that's because in the winter of 2019-2020 we didn't have a lot of influenza, so actually we had less death than the year before. So what we did was to take the maximum, the point-wise maximum between the two curves. So basically for each day, we take the maximum between the official death and the differential mortality. And we obtain this curve over here that are the one that we actually analyze. So the first question that we want to answer is we want to understand if we can group the Italian regions based on the shape of these mortality curves. So to do that, we used a methodology that I developed, well, a few years ago, but it just got published a few days ago. So this is a methodology to do a functional clustering, so clustering of functional data, but in a local way. So it is called probabilistic k-mean with local alignment. Local alignment because we really want to cluster curves based on local characteristics. So not looking at them globally, but looking at small pieces within these curves. Okay, so this slide is a bit technical, so I'll try to make it simple if I can. The idea here is that in functional data, we can have many different types of groups and of clustering. So the most simple one is this case over here. This case over here, in which we have two groups of curves. The curves are aligned, so we only need to find the two groups. But I told you that in functional data, we often have a problem of alignment between the curves. So here we have misaligned curves. So again, only two shapes, but they are misaligned. So, an additional layer of complexity. We can also have curves that differ, that they have, two groups only in a small part of the domain. And finally, we can have curves that differ only locally, but with this local part that is misaligned. So my methodology can deal with all these this type of clusters at the same time, and it can also deal with this problem of finding functional motifs. That is basically finding small patterns, small shapes that are repeated, and they can be repeated within the same curve or across different curves. But going back to COVID data, what we did was to use this methodology to cluster the mortality curves. And we actually, we were able to find two groups, the red group that we call exponential pattern, and the blue group that we call flattened pattern. We find these two groups by aligning the curves. So we also obtain the shift between the curves. This shift basically tells us the different timing of the epidemics in the different regions. So especially for the red cluster, this makes a lot of sense. So we see that basically the epidemic started in Lombardia, and other curves that have a similar shape actually were shifted by 1 week. So everything started 1 week after in the other region. One interesting thing is that I told you that everything started in Lombardia and in Veneto, but you can see that they are in two different groups. And actually in Veneto, in the first wave, they actually did very, very well. because they started testing everyone. So they were actually able to flatten the curve. That didn't happen in the second wave. In the second wave, they did very badly. But in the first wave, this is basically what we see here. So we also used a testing procedure to test for differences between the two curves, the two set of curves. So we wanted to see if those two groups were really two groups or not. So in this case, we employed another methodology that I developed a few years ago that is called IW-tomics. Basically, it is a testing technique. So we are performing a hypothesis test to see if there is any difference between two groups of curves. But the interesting thing about this methodology is able to tell you not only, yes, there is a difference, but also there is a difference in this part of the curve. So basically what we see here at the bottom of this figure is that there is a significant difference. So the gray area is where the difference between the two groups is significant. So in the first part of the curves, the difference is not significant, but then we actually have two different groups of curves. The next step was trying to link this mortality with the local mobility and positivity. So we have, again, our mortality curves, but now we have other two sets of curves. We have a set of mobility curve. So these are from Google, and that's the relative change with respect to January 2020. So we can see that the two lines are the lockdown and the closure of all the activities. So we use the grocery and pharmacies because people basically could move only to go to groceries. And basically we see that there was a huge decrease in mobility. Also, I mean, up to minus 70, 80%. And then we also use positivity data. That basically is the number of positive cases out of all the tests performed. Of course, these are not raw data. These are smoothed data. We had one problem. So we actually had to align these mortality curves because of the different time of where the epidemic started in the different regions. So we basically applied the same shift to mobility and positivity curves to keep the timing between the different data sets. And the question was, can we study the correlation, the association between mortality, mobility, and positivity? So to do that, we used a regression model, okay? The only difference with respect to the usual regression model is that here the response is a curve and the two predictors are curves. So everything is a bit more complex than the traditional regression. So the coefficients are not numbers, but they are surfaces. But it is basically the same thing and the same interpretation of the usual regression. So we applied this model and we basically were able to see that local mobility was actually a strong positive predictor of mortality at its peak. And this is the red part of this beta surface. Late in the epidemic, we had a negative association. Remember that here we are studying only correlation, not causation between the variables. So what we think this negative association mean is that basically in the regions of the south where basically there was no COVID, people just started going around a little bit more. So we think that the causality is exactly the opposite in this case. And about positivity, positivity is a mild positive predictor of mortality. And the late in the epidemics is actually a strong positive predictor. One last step. Here we didn't take into consideration the fact that there are many differences between the regions of Italy. So basically, we collected other data for covariates that can describe these differences between the different regions. So for example, we were looking at the percentage of over 65 years old, the percentage of diabetics, also the ability of hospital to act as a contagion hub, or how big schools are, because all those things can affect the epidemic. So here we employed a methodology, again, for functional data developed by my collaborators. And basically what we did was to try to select among these variables. So we considered the relationship between all these 12 covariates and our mortality curves. And at the end, we were able to select only five important variables that are the adults per family doctor, the number of beds per hospital, the number of students per classroom, the average number of people per household, and the average employees per firm. So we included these variables in the previous model, and basically the conclusion that we had before didn't change. So even controlling for differences between the different regions didn't really change our conclusions. Okay, so to conclude this part of the work, we use functional data analysis methods, a lot of different functional data analysis methods, to study the first wave of COVID-19 in Italy. We were able to prove a strong lagged association of mortality with mobility and positivity. And in particular, mobility can work as a key modulator of COVID-19 spread, and positivity can work as a monitoring variables because they are positively correlated to mortality. Also, we were able to see that distributed primary health care can mitigate mortality, while big hospitals, big schools, and big workplaces can actually work as a contagion hub. If you are interested in seeing more results, we published the results of this project on scientific reports, so you can find much more details there. Currently, we're still working on a second phase of this project. So we're trying to switch in from the 20 regions to 107 provinces in Italy to try to look a bit more in detail what happened in the different provinces. And also we're trying to look at the first and second waves at the same time. Well, here you can see some preliminary results. So one interesting thing is that the provinces here in Lombardia that did extremely bad in the first wave, actually in the second wave did very well. We don't know exactly if it is because all the weak people already died in the first wave, or if it is because people were more careful, because they already had a very bad wave. So at least if I talk to my family that lives in exactly here in this red area, well, apparently people were a lot more careful. So they were following the rules way more carefully during the second wave. But well, yeah, we're still working on this, and we hope to finish this project soon. I don't know if I should take question on this first part now, or if I should go for 10, 15 minutes on the second part. Can I move on to the second part? Okay.
Moi, j'aurais tendance à penser de continuer, ouais.
Okay. Okay, the second part is about omics sciences. So here, well, a lot of collaborators Francesca Chiaromonte is also in this work. Katerina Makova, she's a biologist from Penn State, and well, many other collaborators here. So in the past, well, at this point it is probably 20 years, high throughput sequencing technologies actually allowed very fast and cheap sequencing of the DNA. So the result is that we have a bunch of different techniques that can generate a bunch of different types of omics data. So just to give you some example, if we want to study the relationship between, if we want to study the interactions, between the DNA and the different types of proteins, we can use, for example, ChIP-seq, or we can have RNA-seq to study the transcription of genes, or we can just have whole genome sequencing or exon sequencing to study the mutation that we have in the genome. So all this technology generates a huge quantity of omics data that have some characteristic in common. So they are very complex, very high dimensional. They are usually very, very noisy. So my approach to study this data is to use functional data analysis when I can. And basically here, the idea is that many of these omics data can be represented as curve over the genome. So with this data, we have a lot of problems that can be dealt with functional data. So first problem is that usually we have correlation among the neighborhood positions. And in this case, with functional data analysis, we can actually exploit this correlation because we can ask for smoothness of the signals along the genome. So basically the ordering of this genomic position, one after the other, is embedded in the curve itself. Also, we have a lot of missing values usually, and we can deal with that a little bit with smoothing. So with smoothing, we can fill some of these missing data. And finally, the noise. We have a lot of noise, but also the noise is complex, so it is not very easy to model the noise. And functional data methods usually are not non-parametric, so they allow us to deal with this noise in a non-parametric way. So there are actually three main research directions in which functional data analysis has already been used in omics studies. One is the shape of the epigenome. In this case, the functional data are, the signals, for example, of ChIP-seq. And these are really to study the epigenome. We have shapes of the phenotypes in which we actually try to find a correlation between, for example, SNPs or other omics data with some phenotype. For example, here with the shape of the face of people from different ethnicity. And we have the shape of the genomic landscape. And I'm going to tell you more about this. So by shape of the genomic landscape, I mean that we can use genomic features to have a detailed map of the genomic landscape. So basically, we can look at different features at high resolution along the genome and put all the different omics data together. So we can have many different types of omics data. For example, we can have mutation rates, replication timing, lot of different data. And what we usually do is to contrast regions of the genome that are of interest. So for example, we can look at regions where we have some element of interest versus control regions where we do not have that element. and test for differences between the two, not just looking at the average signal that we have, but really looking at the shapes of these functional data. So I'm going to give you one example in 1 second. So the first example I'm going to talk about is the study of patterns in mutation rates profiles. So from the point of view of the methodology, here we use the functional motif discovery method. So that is the clustering methodology that I was telling you before, exactly the same. From a more biological point of view, here we want to study mutagenesis. So we know that mutagenesis is, that the mutations are not exactly the same everywhere. So mutation rates actually can vary in different regions of the genome. It can vary at different scales, also at different evolutionary times. So my collaborators in a previous work, they were able to segment the genome in 6 different states based on different type of neutral mutation rate. Among these regions, there is this first group that have very high level of insertion, deletion, substitution rates. So these are regions of the genome where we have a huge mutation rate with respect to the average of the entire genome. In this publication, they actually produced this segmentation using very low resolution data. So they were looking at windows of 1 megabases along the genome. So what we did was to consider these hot regions with where we have a lot of mutations, but consider them at high resolution. So we were considering 1 kb windows. We were again computing neutral substitution, insertion, and deletion rate. And basically, the data that you see here are in red, the data from the previous study at 1 megabases resolution, and the green are our data at high resolution. So the question here is that if we can find the shapes that are repeated within hot regions. So basically, we are interested in understanding if within these hot regions, we have regions that are maybe super hot or regions that are cold. Okay, so just looking at them at a higher resolution. So, well, there was a lot of pre-processing, but I can probably just go to the results. So we basically applied the methodology for local clustering and functional motif discovery. And we were able to find 13 different motifs of different length in these hot regions. So these are the shapes that we found that are repeated in different parts of the genome. And there are 2 of these that are particularly interesting. So the first one is the one up here. The one up here, that basically is a super hot regions, because they show 10–20% more substitution rate than the average of the hot regions. So hot regions are already hot, and here we have even more. Another interesting one is this cold one. So here we have 20% less of mutations than these hot regions. So basically, these are normal regions. Okay, so 20% less means the average over the entire genome. And we are also able to see that these motifs correspond to different genomic landscape. So in this heat map, you see a lot of different omics data on the y-axis, and we have the motifs on the x-axis. Basically, red means that this particular omic data is highly expressed in these particular motifs. Blue means lower than expected. White means no significant difference. So I don't want to go into the details, just saying that, that each motif has a specific genomic landscape. Okay, I will go very quickly to another application. In this case, we are again using omics data, but the objective here is to try and understand if non-BDNA can impact the polymerase speed and the error that the polymerase make. So BDNA is the usual double helix that we all know, but there are many regions of the genome where we can actually have very weird three-dimensional shapes of the genome. For example, these G quadruplexes are particularly interesting because basically we have a lot of Gs and the Gs band together and form these very complex three-dimensional structures. So our hypothesis is that whenever we have such a three-dimensional structure in the genome, the polymerase needs to work more to actually be able to go through the genome and replicate the genome. So we wanted to see if the polymerase speed is slowing down, and also if the polymerase makes more errors, so more mutations. So here we used data from smart sequencing, PacBio sequencing. This is sequencing technology that doesn't give us only the sequence of the genome, but it also gives us the interpost duration. So basically, how much time the polymerase in the machine takes to go from one base to the next one. So basically, this is a measure in the machine, of course, not in the living cells, but in the machine, it is a measure of the polymerization speed. So what we did here, well, we took our genome, we selected the regions of the genome where we had a particular type of motif that could form these weird three-dimensional structures. For example, here we have a G quadruplex. Then we took regions of the genome that should form the normal double-stranded DNA. We put them all together, and basically we looked at the curve of this interpost duration in the two groups. At this point, we have two groups of curves, and we can test for differences using the testing methodology that I was showing you before. So the objective is to try to understand if there is a slowdown of the polymerase or not. The answer is yes, there is a slowdown. So I will skip the one on the left, but the figure on the right basically shows you this interpost duration in the control regions, so the usual double-strand regions, and those are in blue. And in red, we have the interpost duration for G quadruplex. And here in the middle, we see where these motifs are. So these motifs have different lengths. So we basically align them so that their center is at the middle of this window. So basically, in the middle, we have a lot of G quadruplex. And then here we only have some, so the longer one are still here. At the bottom, we see the other strand of the DNA. So the interesting thing about G quadruplex is that they form where the Gs are. So they form only on one of the two strands of the DNA. So what we see is that there is a huge increase in time where the G quadruplex is, but we do not see the same shape on the opposite side. The next step was trying to understand if this slowdown also corresponding to more errors in the genome. So basically, here we studied small scale variation in nucleotide substitution frequency, looking again at the places where we have this non-B DNA. we checked different type of substitution frequencies. So we look at diversity and divergence. So we looked at intraspecific variation and interspecific variation. We used again the same methodology, and we saw that where we have these non-BDNA, we actually have an increased number of substitutions. So in this figure, for example, we can see all the G quadruplexes in blue, and in black we have the controls. So we see that there is a huge increase. And we also see that the increase is not the same in stem and loop. So basically, you see that the non-BDNA is this weird shape. So we have a stem and we have also this loop over here. And there is also a difference between these two. And we saw the same also for different type of non-BDNA. For example, here we have zDNA, and we see again an increase, but the shape is pretty different from what we saw for G quadruplexes. So to wrap up this part of the talk, we used the functional hypothesis test to contrast genomic regions and prove that we have a decreased polymerization speed at non-BDNA and also an increased mutation frequencies at non-BDNA. And the functional data analysis tool actually allowed us to really consider the curves and being able to say not only, yes, there is a difference, but also the difference is here. But then if we look a little bit outside of the motif, so a little bit outside of the G quadruplex, the difference is not there anymore. And thank you very much. I would be very happy to answer any question.
Merci, Marzia. Est-ce qu'il y a des questions? Ben, je ne peux pas voir dans la salle en personne. Je ne sais pas si Marzia, tu vois des mains levées, mais sinon, je sais pas, en ligne.
Oui, il y a une question ici.
OK, je vais te laisser la...
Je me demandais, dans vos modèles sur les courbes de COVID, là, vous avez mentionné que vous avez shifté le mobility data du même nombre de jours que le shift des courbes. Je me demandais, est-ce que vous allez considérer ajouter un lag period? Parce que, admettons, la mobilité d'aujourd'hui prédirait, je pense, mieux dans 10 ou 14 jours, admettons?
Oui, donc ce n'est pas nécessaire.
Okay.
Donc la belle chose de ces méthodes-là, sont que les bêta, donc les coefficients, sont des surfaces. Donc, on va voir la relation entre chaque temps de la réponse et chaque temps du prédicteur. Donc, ce n'est pas nécessaire. Donc, on va voir les legs après qu'on a ajusté le modèle, quand on regarde les bêta.
Ça va être intégré dans l'estimation de bêta.
Oui, exactement.
Est-ce qu'il y a d'autres questions?
Oui, ici.
Dans les premières applications que vous avez montrées, il y a des choses qui me faisaient penser à des analyses de trajectoire. Est-ce que vous savez de quoi je parle de ça? Base modeling, je pense.
Oui.
Comment ça se compare, les deux méthodes, savez-vous? Est-ce qu'il y a des avantages et inconvénients? Non?
Je ne sais pas exactement. Les avantages de l'analyse des données fonctionnelles, c'est qu'on fait tout vraiment de façon non paramétrique. Donc, je pense que c'est plus facile de comprendre les relations quand on ne doit pas vraiment avoir un modèle pour modéliser l'erreur ou pour modéliser les relations entre les variables. Donc, je pense que ça, c'est une des choses positives. Un inconvénient, c'est que dans les données fonctionnelles, on ne regarde pas la direction du temps. Donc, on pourrait avoir aussi le futur qui a une influence sur le passé. Donc ça, c'est l'inconvénient. Ouais. Ouais.
Est-ce qu'il y a un nombre de points minimum? J'imagine que plus que, parce que vous, ce que vous avez analysé, il y a plusieurs points, mais admettons, est-ce qu'il y a un nombre de minimum pour que cette analyse soit valable?
Non. Non et oui, disons. Si on a beaucoup de points, bon, ça va être plus facile d'estimer la courbe. Et on peut estimer chaque courbe de façon séparée. Si on n'a pas beaucoup de points, comme dans un des graphiques que j'avais montré au début de la présentation, dans ce cas-là, on ne peut pas vraiment estimer chaque courbe, mais on doit mettre les courbes ensemble. Donc, il y a des méthodes d'analyse de données fonctionnelles pour essayer d'estimer les courbes quand on a des données sparse data. Donc, dans ce cas-là, on utilise l'idée que toutes les courbes ont la même forme pour estimer les courbes, même si on n'a pas vraiment beaucoup de données. Ouais. Ouais. On a une autre question ici.
Vous avez parlé dans l'application OMI qu'il fallait tenir compte de la corrélation entre les différentes localisations proches sur le génome. Il n'y a pas ça dans les autres applications où votre axe des X, c'est le temps, donc s'il y avait de la corrélation?
Oh oui, c'est la même chose. Oui, c'est la même chose. Donc, dans le temps, on a une corrélation, oui, bien sûr, entre des instants qui sont près l'un de l'autre.
Okay,
donc, oui, c'est la même chose, oui.
OK, mais s'il n'y a pas d'autres, moi, je ne vois pas de questions en ligne. J'aimerais dire un gros merci à Marzia pour cette présentation, super intéressant. Puis merci à tout le monde pour votre participation. Je trouve ça, c'est vraiment un domaine, en tout cas pour moi, très, on va dire, des méthodes distinctes ou nouvelles que je connais extrêmement peu. Donc, c'est intéressant d'apprendre d'apprendre sur ces méthodes. Donc, je mettrai fin à cette à ce webinaire. Puis c'est ça. En vous remerciant tous de votre participation.
Merci. Bonne journée à tous.
Au revoir.
Okay. Merci.